I'm excited to talk about quite a different aspect of insect populations and communities, which is their microbiome. So interactions with other organisms are a really important aspect of biodiversity, as well as insect biology. So we often think about plant-herbivore interactions or predator-prey interactions, but interactions with microbiome can be just as important. By microbiome, I mean, or researchers typically mean, the community of microorganisms that can be found living together in a given habitat. So microbiomes are critical to biology of most organisms. Currently, research focuses on humans because microbiota are so critical to our health. But they are not less important, and often much more important, for diverse insects. So those microbiota of diverse insects are very taxonomically diverse themselves. They are also very diverse functional. So we distinguish many different functional categories of insect microassociations that differ in their association stability, tissue localization, in how essential they are to insect hosts. They differ also in the types of transmission.
So some microbes have been transmitted strictly maternally from mothers to offspring, sometimes for hundreds of millions of years. Other microbes are acquired from environment each generation. And then there are some microbes which can move across species, and after establishing in a new species, perhaps start transmitting again from mothers to offspring across generations. Those different functional and taxonomic categories of microbes have many important effects on their insect hosts. The most important categories of effects are nutrition, defense, and reproduction. We'll provide some examples. So a great example of nutritional symbionts are specialized microbes that live inside the tissues of hoppers, so leaf hoppers, plant hoppers, and cicadas, as they provide essential nutrients that are lacking in their diet of plant sap. So again, the insects completely cannot survive without specialized microbes. Symbionts can have a wide range of protective effects, and there are lots of many studies which show them in different systems. So symbionts can protect against pathogenic fungi, against viruses, against parasitic wasps, or parasitoids more broadly, against predators. They can also protect against a range of environmental pressures, for example, against high temperatures, or against drought, or against toxic chemicals, including pesticides. Then symbionts can have a wide range of effects on insect reproduction. So they can feminize genetic males, induce parthenogenesis, which is essentially clonal reproduction, kill males, or cause an incompatibility between infected and non-infected insects or lines. And all those mechanisms help improve microbial transmission within insect populations. And again, many different symbionts can have different effects at the same time. So a great example, I would say, like maybe the hottest topic in insect microbiome studies are mosquitoes. It was shown that introduction of the symbiotic bacterium Wolbachia into mosquitoes have blocked their ability to transmit deadly viruses such as dengue or Zika. So now, because of that, Wolbachia is used extensively to control disease around the world in different countries. But once introduced into population, thanks to its reproductive effects, Wolbachia can persist through reproductive manipulation. So once we know those effects, they can really be put to good use. I already started hinting that those symbiont effects can be detected at many different levels. So not just at the level of individuals, but also at the level of populations. Symbionts can affect evolution of their hosts, and they have potential to affect entire multi-species communities through cascading effects among different interacting species. One of the most clear effects of this symbiont population level effect was described from Australian Drosophila simulans. As you can see here, this red strain of Wolbachia invaded Australian population at around 2004, and then in less than a decade, it has swept through continental scale populations of the species. Again, having major effects on also genetic structure of the flies. Another recent example is from Chinese whiteflies. It was shown that between 2019 and 2021, whitefly populations were invaded by this pathogenic fungus that is really deadly for whiteflies. And simultaneously with the spread of fungus, the spread of protective symbiont that dramatically decreases the costs of infection, greatly and reduces transmission of the fungus. So was observed. So fungus spread and protective symbiont has spread, again, very substantially, reducing population susceptibility to this important environmental pressure. So those and many other examples argue that symbionts should be considered as really important parts of insect evolution. I am thinking about symbionts as agents of this very rapid response and adaptation to environmental challenges and opportunities. And they do include many of the pressures that do drive global biodiversity declines, including climate change, pollution with toxic chemicals, or invasive species. Still, despite their importance, symbionts are very poorly characterized outside of model systems that do include humans, and then just a handful of well-known species. So, how to study insect microbiota as a part of those big biodiversity surveys? I would say that one of the really essential things is a large collection of comprehensively sampled communities, and so I was fortunate to work with Insect Biome Atlas collections to get very diverse Swedish insects from all over the place. Once we have those bottles full of insects, those community samples, we can extract the DNA from the entire multi-species bulk sample. Or another approach that I have been preferring is sorting the sample somewhat. So not necessarily by taxonomic experts, because this can be, as you have already heard, very slow and time-consuming, but sort them sort of roughly so that we can extract much of the biodiversity present in the sample, and then extract the DNA from insects individually. Also, we are now finding that we can also sort insects after mild lysis, and then also use them for microbiome or individual characterization or individual microbiome work. Once we have the DNA, whether from bulk samples or from individual insects, we can apply one of several methods. So you, of course, know about CO1 metabarcoding, which teaches us about insect diversity, but it can also provide valuable information about certain microbes, such as Wolbachia, which is the most broadly distributed of insect symbionts. We can also sequence not just insect, but also microbial marker regions, getting more precise information about microbial diversity. Or, as you have also heard about already, we can just sequence the total DNA extracted from such sample and obtain detailed information about identity, phylogenetic relationships, and functions. Yes, but there is a caveat, so the cost increases a lot if we go from metabarcoding to metagenomics. But on the other hand, throughput is much higher for those cheaper methods. And the great thing is that we can combine the different methods. So we can start very broadly to get a broad sense of the community structure, before then perhaps pinpointing on specific systems and characterizing them in detail. Using such combined approaches, we can start addressing questions at really massive scales. For example, asking questions about microbiota distribution and the dynamics across an entire range of an insect clade, microbiota structure and transmission within this diverse multi-species community, or about microbial importance, influence on insect ecology and climate change responses. We have been addressing several different questions in different systems. This is very much in the works. In particular, these biggest analyses of microbiome diversity patterns from across the Swedish insect diversity will take more time. But I wanted to share some of the results for one of the systems. We have good data for Swedish scuttleflies, which is one of the dark taxa. So one of those hyper-diverse insect clades that are very poorly characterized. Yeah, so thankfully, thanks to contribution of Emily Hartop, we got quite a good, we got lots of help with figuring out who the different scuttleflies are or how to work with those complex data. And then we used a collection of close to 2,000 flies representing well over 100 species as revealed by individual CO1 barcoding. One of the very striking aspects of our results was that bacterial abundance varies dramatically among insects. So here we sorted insects by species and then by sex and then by decreasing bacterial abundance. And what I found really striking is that there are up to 10,000-fold differences in how much bacteria there are, even among individuals of the same sex and species. Yeah, we also observe systematic differences between males and females in how much bacteria there are. Here, for the insects sorted the same way, we plotted relative abundance of dominant bacterial clades. And I think that you can see quite clearly that the patterns are not random. So variation in microbial abundance is at least partially explained by community structure, and both are explained by host species and sex, despite the huge variation. Those patterns do correlate with some of the bacterial functions that we can predict. So here we plotted, for 5 most abundant species, we presented information on prevalence of infection and abundance of bacteria. And so, for example, for Wolbachia, in species 1, we found that about 1/4 of males are infected, but 3/4 of females. And infected females tend to have about 30 times as much bacteria as males. So those patterns are strongly consistent with some types of reproductive manipulation. We observe similar patterns in different microbial categories, so in Wolbachia for different species, for some other microbes. We can also obtain genotype level or clade level information on symbiont specificity. So here we plotted the distribution of Wolbachia clades across the 4 species. And you can see, again, non-random patterns. But we do get much more resolution by looking at genotype level interactions. So presence of Wolbachia genotypes in different Wolbachia species. Again, lots of variation. Some genotypes seem to be shared. Genotypes tend to associate typically with one variant, and so on. But we can follow up with methods that provide higher resolution, such as metagenomics. And so here, for about 80 specimens representing different insect microbe combinations, we have generated those multi-level phylogenies, describing entirely new unknown clades of Wolbachia. And then, when comparing the insect phylogenies, symbiont phylogenies against insect phylogenies, we were able to learn about specificity and transmission patterns. So By combining those methods, for large numbers of wild-caught insects, we can obtain insect identity information, microbial abundance and the distribution patterns, information on differences among species, populations, and sexes. We can reconstruct functions from population data. We can perhaps identify entire new microbial clades, learn about microbial transmission across the community. And then, well, that work is upcoming, but we can get much more information about functions and the mechanisms of symbiosis. So with that, I hope I convinced you that microbial symbionts can play critically important roles in biology of their host organisms. We do have tools to comprehensively describe those patterns at ecosystem level. And in the era of dramatic anthropogenic changes and biodiversity declines, it is really important to understand those patterns, describe them comprehensively. So I wanted to thank my research group in Krakow, Insect Biome Atlas team, Station Line team, well, all of you who contributed by gathering invaluable material, and funders. Thank you.